In this section, we will talk about the skeletal muscle structure. A skeletal muscle cell is called a muscle fiber. A delicate connective tissue, called endomysium, covers each muscle fiber. Muscle fibers are grouped in bundles, called fascicles. A sheath of tougher connective tissue, called the perimysium, encases the fascicles. Still, another layer of connective tissue, called the epimysium, surrounds the muscle as a whole and binds all the muscle fibers together. Connective tissue, called fascia, surrounds the muscle outside the epimysium. Deep fascia lies between muscles, while superficial fascia resides just under the skin. Tendons and aponeuroses are so strong that they rarely break, even by forces strong enough to break a bone or tear a muscle. They can, however, be pulled away from a bone. Moving towards structure of muscle fibers. Because of their long thread-like appearance, muscle cells are called muscle fibers. Unlike other cells, muscle fibers have multiple nuclei pressed against the side of the plasma membrane. Furthermore, even though muscle fibers are extremely thin, they contain a complex interior, just like other human cells. The plasma membrane surrounding each fiber is called a sarcolemma, whereas the cytoplasm of the cell is called sarcoplasm. Long protein bundles, called myofibrils, fill the sarcoplasm. Myofibrils store glycogen, which is used for energy, as well as oxygen. Sarcoplasmic reticulum is the smooth endoplasmic reticulum of a muscle fiber, surrounds each myofibril. This is where calcium ions are stored. A system of tubules, called transverse tubules, extend across the sarcoplasm, formed from inward projections of the sarcolemma. The T-tubules allow electrical impulses to travel deep into the cell. Myofibrils consist of even finer fibers, called myofilaments. There are two types of myofilaments, thick and thin. Thick myofilaments are made of a protein called myosin, while thin myofilaments consist of a protein called actin. The arrangement of actin and myosin gives skeletal muscle its striated appearance. Let's discuss the structure of actin and myosin in detail. First is the thick filament. Each thick myofilament consists of hundreds of myosin molecules stacked together, with the myosin heads facing outward. The myosin molecule, which makes up thick myofilaments, is shaped like a golf club with a globular head and shaft-like tail. Other is the thin filaments, consisting of two chains of the contractile protein actin. Thin myofilaments look like a string of beads. Entwined with the actin are two other proteins, tropomyosin and troponin. Moving towards structure of myofibril. The thin and thick myofilaments stack together, one alternating with the other, to form myofibrils. They do not completely overlap. Instead, they're arranged in a type of latticework to form units called sarcomeres. A plate or disc called a Z-disc or Z-line serves as an anchor point for thin myofilaments. The section between the Z-discs is called a sarcomere. This is where muscle contraction occurs. In a relaxed muscle, the myosin, which is thick filament, and actin, which is thin filament, lie side by side, partially overlapping. The myosin and actin are completely detached from one another. Contraction occurs when the myosin heads latch onto the actin myofilaments. This forms what is known as a cross bridge between the actin and myosin. The myosin heads latch onto and release the actin repeatedly, creating a series of power strokes that propel the actin myofilaments forward toward the center of the sarcomere. Because the actin myofilaments are attached to the Z-discs, they pull the Z-discs closer together, shortening the sarcomere. As the sarcomere shortens, so does the myofibril and the entire muscle. This is known as the sliding filament model of contraction. This was all about this section. In our next section, we will understand contraction of muscles in detail.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.